Hello and welcome to Bio40 Pre-Lecture 11. Let's review DNA replication and talk about how the process differs in prokaryotes and eukaryotes. Last time we began by discussing the most important enzyme, the polymerase, and building from there. This time I want to review the process chronologically. Replication begins when an initiator protein binds a specific sequence of DNA called the origin of replication. The binding of the initiator causes a small distortion in the DNA that helps recruit the enzyme DNA helicase. DNA helicase unwinds a double helix and separates the strands, leaving unpaired strands that can serve as templates for DNA polymerase. This is one of the steps in replication that requires energy in the form of ATP. The spot where double-stranded DNA separates into single strands is called the replication fork. It will move down the DNA as a helicase advances. Ahead of the replication fork, topoisomerase relaxes supercoils in the DNA. Supercoils are overwound DNA that could block the fork from advancing. Topoisomerase also requires energy in the form of ATP to function. So we've got our replication fork going, but we need to worry that the single-stranded DNA will come back together, or anneal, before the polymerase has a chance to synthesize new strands. Single-stranded binding proteins prevent this from occurring. There's one more step that must occur before DNA polymerase can synthesize the new strands. Because DNA polymerase can only extend existing chains and not start new ones, we need something that can start off the new strands. That something is primase, an RNA polymerase that can synthesize RNA primers, or short chains of nucleotides, without building on any pre-existing chain. This is another step that requires energy, but here the energy is provided by the RNA building blocks, NTPs. The RNA primers provide the 3' hydroxyl groups that the DNA polymerase needs to start building the DNA chains. And just as the NTPs gave the energy required for the primase, the dNTPs that serve as the building blocks for the DNA chain are also the polymerase's energy source. As the DNA polymerase extends the chain, it sometimes makes errors, pairing the wrong base with the template strand. Some polymerases have 3' to 5' X-nucleus activity that can excise the incorrect base so that it can be replaced with the proper one. Errors can still get past these polymerases, but at a much smaller rate than with polymerases that don't have this activity. There's another big consequence of DNA polymerase's limitation of only being able to add nucleotides to existing 3' hydroxyls. This means that synthesis can only occur in the 5' to 3' direction. We can see the complications this causes in this image. The replication fork is advancing to the left of the screen here. For the top strand, the leading strand, the polymerase just keeps moving in the direction the fork is moving, and there's no problem. But with the bottom strand, the lagging strand, there is a big issue. Synthesis begins at the fork and moves away from it. So when the fork advances, the process needs to start all over again. Primase adds a new primer, and DNA polymerase extends the chain away from the fork until it bumps into the primer from the previous round. This results in a series of discontinuous fragments called Okazaki fragments. Certain DNA polymerases have 5' to 3' X-nucleus activity that can remove the RNA primers and replace the sequences with DNA. However, the polymerase that does this is not necessarily the same one that does the general chain extension. With this polymerase activity, the new strand is entirely DNA now. But polymerases cannot seal the gaps in the DNA backbone between adjacent Okazaki fragments. This task is accomplished by the enzyme DNA ligase. Forming these phosphodiester bonds is another step that requires energy in the form of ATP. Now we finally have two unbroken chains of DNA on both the leading and the lagging strands. Sorry to be repetitive, but this is a topic that can easily confuse people, so I want to show the process in several ways. Here's a replication fork in the bacterium E. coli. The initiator has already started the process, and DNA helicase is unwinding the double helix, with topoisomerase running ahead to relax supercoils. Single-stranded binding proteins are stabilizing the single-stranded DNA to prevent it from reannealing. Primase starts off the new strands with RNA primers, and DNA polymerase extends the chains as DNA. In E. coli, the polymerase that does this is called DNA polymerase 3. On the leading strand, the polymerase can just keep extending the same chain running just behind the replication fork. But on the lagging strand, the polymerase starts at the replication fork and moves in the opposite direction until it bumps into an earlier fragment and falls off. Meanwhile, primase makes a new primer at the point where the fork is advanced, and the DNA polymerase can continue a new fragment from there. And the process repeats over and over. In E. coli, DNA polymerase 1 does the job of removing the RNA primers and replacing them with DNA. And DNA ligase seals a nix between the fragments. And that's replication. Now, these still images are nice and all, but for some people, video can be really helpful. I don't have any animation skills, but luckily other people do, so I recommend checking out the other video that I've posted on the lessons page. Here's a brief summary of the proteins involved in replication, with the energy sources highlighted in red. This isn't meant to include all the knowledge you need to know about DNA replication, but it's a handy summary of many of the major points. Now for a few issues about replication that we haven't discussed yet. In explaining the basics of the process, we tended to look at one replication fork moving in one direction. But in reality, replication is bidirectional. There are two replication forks moving in opposite directions. And each fork has a leading strand and a lagging strand. It's interesting to note that the leading strand of one fork is a lagging strand of the other fork, but synthesis is always occurring 5' to 3'. Next, let's look at some differences between prokaryotes and eukaryotes. E. coli is a typical model organism for studying biological processes in prokaryotes. E. coli has one chromosome, and that chromosome has a single origin of replication. The site is called ORI-C, and it consists of 4 repeating sequences, 9 base pairs in length, and 3 repeating sequences, 13 base pairs in length. DNA A, the initiator protein in E. coli, binds the 9 base pair repeats. This binding distorts the DNA and causes the strands in the 13 base pair repeats to separate. The 13 base pair repeats are AT-rich, and you'll recall that AT base pairs are only joined by 2 hydrogen bonds, as opposed to the 3 in GC pairs. So it's relatively easy to separate the strands in this region. This distortion helps recruit the DNA helicase to start the replication forks, followed by primase and the rest of the proteins we've been discussing. Unlike the linear chromosomes we've seen in animals, bacterial chromosomes are typically circular. So the replication forks proceed all the way around the circle until they meet at a termination site at the opposite end of the chromosome. At this point, the daughter chromosomes are joined at the termination site, but a topoisomerase separates them. This is not the same as the topoisomerase that relaxes supercoils, though it does function in a similar manner. We can contrast this with eukaryotic chromosomes. Most eukaryotes have multiple linear chromosomes, and each chromosome has several origins of replication. Replication begins at multiple points, and the forks move outward until they reach the end of the chromosome, or encounter a replication fork moving in the opposite direction. Having multiple origins is important because eukaryotic chromosomes are so large. While the E. coli chromosome is under 5 million base pairs in length, the largest human chromosome is nearly 250 million base pairs. One big issue with linear chromosomes has to do with their ends. The ends of linear pieces of DNA tend to stick together, but we don't want our chromosomes fusing like that. So we have structures at the end of our chromosomes called telomeres that prevent this from occurring. But as our cells divide and chromosomes replicate, our telomeres can shrink. Let's look at why that is. We talked before about how the RNA primers needed to start synthesis are removed and replaced with DNA. And while that's mostly true, there's a problem when it comes to the very 5' ends of the newly synthesized strands. Primers in the middle of the DNA can be removed and replaced by extending the chain of a neighboring fragment. But after we remove the primers at the ends of the DNA, there's no 3' hydroxyl to build from. We're left with these gaps. And after the next round of replication, there will be a bigger gap. And so on and so forth until the telomeres are lost completely and nothing is protecting the chromosomes from fusing together. But our cells have a way of dealing with this issue using the enzyme telomerase. Our telomeres consist of hundreds to thousands of repeats of the base sequence TTA-GGG. The figure we're looking at has repeats of TTG-GGG because it's actually from the protist tetrahymena. Using this system, let's imagine a situation where after a round of replication, there is now a gap of 12 bases or 2 repeats. Telomerase will find and bind to this gap. Telomerase is what we call a ribonucleoprotein. It consists of a protein, a DNA polymerase, and a single-stranded RNA template. This RNA has a sequence that allows it to base pair with the telomere repeats. But not all of it pairs. There's an overhang that can serve as a template to extend the top DNA strand, which does have a free 3' hydroxyl. After this occurs, the telomerase shifts, and again provides a template to further extend the top DNA strand. Polymerases like this, that synthesize DNA using RNA templates, are known as reverse transcriptases. After the second round of DNA synthesis, a whole new TTG-GGG repeat has been added to the top strand. Now if the same 12-nucleotide primer is used to initiate synthesis on the bottom strand, 6 new DNA nucleotides can be added. When the primer is removed, there's still a 12-nucleotide gap, but the whole thing has been shifted over by a full repeat. Now we actually don't express telomerase in our somatic cells. It's important in germline cells that give rise to gametes, but our somatic cells are supposed to age and die. The lack of telomerase is an important precaution against uncontrolled cell growth, which is cancer. So improper activation of telomerase in somatic tissue is a common step in the progression of cancer. That does it for our overview of how replication normally works. But there's one more aspect I want to cover next time: how we've adapted replication to create important lab technologies.